I would like to introduce our webinar speaker, Dr. Volker Stadler. Dr. Stadler is the CEO and co-founder of PEPperPRINT. He graduated in chemistry at the Heidelberg University and received his PhD in physical chemistry. At the German Cancer Research Center in Heidelberg, Dr. Stadler contributed to the development of PEPperPRINT's new peptide microarray technology, which was published in Science and Angewandte Chemie. From 2009 on, he was responsible for the translation of the peptide microarray technology into a commercial product and service business and launched PEPperPRINT in 2010. PEPperPRINT was awarded with various innovation and business awards and reached a break-even in 2014. In 2016, Dr. Stadler was appointed to the executive board of the BioRN Biotech Cluster Rhine-Neckar. So the title of the presentation will be Antibody Validation and Antigen Discovery by Combining Protein and Peptide Microarrays. So once again, please use the question pane to type any questions that may come up. And again, we will share the slides and the post-webinar file on our website. Volker, I'm handing the cursor over to you. Please take it away.
Okay, thank you. Thanks for the introduction, Carsten, and thank you all for joining the webinar. The title has already been given by Carsten. It's about antibody validation by combining protein and peptide microarrays, and this approach can also, of course, be used for antigen and epitope discovery. For those who do not know us, a short introduction. The company is founded and based in Heidelberg, Germany, operative since 2011. We are completely privately held independent company with a quite experienced team of executives and a proven track record. We have, since 2014, an ISO 9001 certified quality management system, presently around 25 employees, and are working with customers all over the world. We had projects with, I think, presently 9 of the top 10 pharma companies. Our core technology, which is fundamental for the approach we are introducing, is a laser printing technology to generate peptide microarrays. Instead of working with tiny droplets as in spotting technologies, we are working with small amino acid toner particles that are printed with a custom peptide laser printer. You can see right on bottom. Instead of colors, like in a typical office laser printer, the amino acid toners contain the different amino acids, and we have 24 printing units in our printer, which corresponds to 24 different amino acids that can be printed at once. We print the toners directly onto glass slides where they are melted to initiate peptide coupling, and the rest is standard ethmochemistry. We wash away residual toner material, deprotect the internal protection group, and print the next layer of amino acids. And this is repeated until a complete peptide microarray is synthesized. It can be either a linear peptide microarray or a microarray based on cycle-constrained peptides for conformational studies. The benefits of the technology is the low material consumption, what translates directly into costs. And we have very high spot densities with around 1,000 different peptides a square centimeter, a high peptide quality based on routine double coupling steps, and most importantly, the laser printing enables us to access a digital printing flexibility in fast production times. For us, it makes no difference if we print 100 times the very same or 100 different microarrays. And this also enables us to provide a variety of different peptide microarray formats, ranging from highly multiplexed formats on the right, with 16 array copies on a standard glass slide and 270 peptide features per array copy, to discovery formats with around 75 different peptide features on a single chip. We have, as you can see on our website, a number of standard peptide microarrays. The strength of our technology is actually the possibility to provide all kinds of custom peptide microarrays. Apart from antibody validation, what is the main topic of the presentation today, we are working in number of disease areas and serological applications in cancer research, infectious disease research, autoimmune research, and also in allergies. Again, the main benefit is the unlimited flexibility of the peptide microarray platform and the possibility not only to investigate linear epitopes, but also conformational peptides by applying the cyclic constrained synthesis. So antibody validation, what can we provide? First of all, of course, linear and conformational epitope mapping services from single to multiple antigens with amino acid resolution. That is, you can really get the highest detail of epitope information possible. Once an epitope is known, we can proceed with epitope substitution scans, where we exchange all amino acid positions of an epitope by the 20 main amino acids, which is similar like an alanine scan, but not only with alanine, with 20 amino acids to investigate essential, conserved, and variable amino acid positions, and two different approaches for the profiling of antibody cross-reactivities with either the human epitope microarray or the HuProt protein array. Short introduction to linear epitope mapping. This is probably straightforward. An antigen is translated into overlapping peptides that are printed onto a microarray. Such a microarray can be is incubated with a target antibody, followed by staining with fluorescently labeled secondary antibodies. Readout with a microarray scanner usually gives access to a row of double spots. As you can see on the left, for a monoclonal or a polyclonal sample, these are raw microarray scans. And once you have the peptides that respond with the antibody, you can narrow them down to the consensus motif, which corresponds to the epitope of the antibody. This is not only possible with linear peptides, as mentioned before. We can also do this for a conformational epitope mapping. And this approach was benchmarked with rituximab, a chimeric monoclonal anti-CD20 antibody. We generated two different microarrays, CD20 as linear or cyclic constrained overlapping peptides with different peptide length, 7, 10, and 13 amino acids, and incubated both microarray variants with the monoclonal antibody. And on top you see that the linear peptides do not give any response with rituximab. It's well known that rituximab binds against an extracellular 15 amino acid loop of CD20. But with the cyclic constrained peptides, we're able to identify nice responses that increase in intensity from the 7 to the 10 to the 13 amino acid peptides, what simply reflects an approach to the natural conformation of the extracellular CD20 loop. And we were able to identify the epitope of rituximab with also amino acid resolution. And the data fits nicely to previous crystal structure analysis. Some research highlights before we go to the actual topic of the presentation. The group of Lancia-Becker et al. Published last year in Nature Medicine a paper on the identification of neutralizing malaria antibodies by using our epitope mappings. A group of the University Hospital in Heidelberg did proteome-wide Zika virus epitope mappings with patient sera from Brazil, published this data and also patented half of the work. It's a nice biomarker that exclusively recognizes Zika virus antibodies, but does not cross-react with dengue virus antibodies. Janssen Diagnostics did some proteome-wide epitope mappings with a tropical pathogen. And a group at the Icahn School of Medicine at Mount Sinai applied our conformational epitope mappings for the identification of neutralizing antibodies in CMV infections. Now we come to the actual topic of the presentation, which is the antibody cross-reactivity analysis, based on a combination of two microarray platforms. The idea was to initially apply a broad cross-reactivity profiling of antibodies with the HuProt Human Proteome Microarray, which is generated by CDI Labs in the US and available in Europe by Cambridge Protein Arrays. The Human Proteome Microarray contains around 20,000 different human proteins. And once we have the hit proteins of the protein microarray, we translate these hit proteins into multiplexed epitope mappings by using our peptide microarray platform, on one hand for hit validation and second for epitope identification. In the third step, once we have the epitopes, we can further apply epitope substitution scans by translating the original wild-type peptide into 100 different peptide variants by exchanging all amino acid positions with the 20 main amino acids to investigate conserved, variable, and even essential amino acid positions. To test this approach, we started with a commercial mouse monoclonal antibody against human transglutaminase 2. The antibodies available at Cidira GmbH in Darmstadt, Germany. And this antibody has been validated by multiplexed epitope mappings against a variety of transglutaminases. And the epitope was identified, it's shown here in the red. And it's known that this antibody does not cross-react with other transglutaminases. Why did we select this antibody? It was simply available in our labs. So we initially incubated a HuProt Human Proteome Microarray with the mouse monoclonal antibody. And what we observed is roughly what can be expected. We observed on the left, you see the microarray scan of the HuProt Human Proteome Microarray. On the right, you see the intensity profile plotted against the whole microarray content. And what we observed is that the strongest response, which is highlighted by the white circle in the microarray scan, is actually directed against transglutaminase 2. But we also observed two weaker cross-reactions against proteins that were not related to transglutaminase 2. In a second step, we translated the protein hits into overlapping peptides for a multiplex epitope mapping with the help of one of our peptide microarrays. We had 12 protein hits translated into 5,000 something different peptides, and incubated this microarray again with a mouse monoclonal antibody. And what we observed is a nice N-terminal epitope that correlated with the known epitope identified before in the validation step. But what we did not observe is any cross-reaction against the other protein hits of the protein microarray. So we had no off-target binding of the antibody against the peptides derived from the protein hits of the protein microarray. The interactions we see on the right of the intensity plot are a result of the second background interactions of the secondary antibody, but do not really play a role with the protein hits of the microarray. In the final step, we took a 15 amino acid wild-type peptide that contains the epitope, which is shown in the underlined amino acid positions, and exchanged all amino acid positions by all amino acids. That can be seen on the left. Below the raw microarray scan, you see the heat map of the microarray scan. And we start with the glutamic acid in the C-terminal position down to threonine in the N-terminal position, and exchanges done for alanine, cysteine, glutamic acid, etc., etc. When we translate this intensity profile into an amino acid plot, which is shown on the right, we normalize all spot intensities of all amino acid exchanges to the intensity of the wild-type peptide, which is set to 100% as a reference value. And that way we can nicely see how conserved, variable, or even essential such an amino acid position for antibody binding is. And what we observed here is that the antibody recognizes an epitope with 7 well-conserved amino acid positions, what makes off-target binding simply less likely. What we can also do is to further investigate this core motif by a database blast with a substitution matrix. We developed a software solution for this as well, but this is beyond the scope of this presentation. So coming from the protein array, we narrowed the response profile down to the epitope and even investigated the conserved character of each individual amino acid position. So this was the mouse monoclonal antibody, but we thought, okay, let's compare it with a slightly different antibody that is also commercially available, and we selected a rapid polyclonal antibody against the same antigen that is provided by Atlas antibodies. On one hand, the Atlas antibodies are known to be highly validated. Here it was done by comparison with the second anti-transglutaminase antibody in immunohistochemistry in Western blot. And it was also claimed that in the ALPAF meeting 2017 that rapid polyclones perform better than monoclonals since they simply recognize more epitopes. So we thought, okay, let's give it a try and compare this antibody with the mouse monoclonal antibody. We incubated another human proteome microarray with the rapid polyclonal antibody against transglutaminase 2, and were quite surprised to see such a strong cross-reactivity profile against a variety of different antigens. As you see on the microarray scan, it's like a heaven full of stars. And surprisingly, what we did not observe is any response against transglutaminase 2, as you can see in the comparison with the intensity plot of the mouse monoclonal antibody. So we were quite surprised by this finding and proceeded again with the peptide microarray with the protein array hits. And surprisingly, in contrast to the protein array, we observed a strong polyclonal response of the rapid polyclonal antibody against a variety, 4 different epitopes close to the N-terminus of transglutaminase 2. However, we were also able to validate a number of cross-reactions of the protein array, albeit at a significantly lower intensity level than on the protein array itself. Compared to the mouse monoclonal antibody, we see, okay, the mouse monoclonal antibody recognizes an N-terminal epitope. It's a C-terminal polyclonal response with 4 epitopes with the rapid polyclonal antibody. So the question was, why have we not seen any response of the rapid polyclonal antibody on the human proteome array? And this is just a part of the microarray data, of the peptide microarray data. This is just the epitope mapping against transglutaminase 2. And to investigate the apparent false negative results on the protein array, we were in discussions with Cambridge Protein Arrays. What happened with the transglutaminase 2 on the protein microarray? And it turned out that the protein microarray did not cover the expected transglutaminase 2 isoform 1 that contains the C-terminal part against which the rapid polyclonal is directed. It contains a transglutaminase isoform 2 where the C-terminal part is missing. So the false negative result of the rapid polyclonal antibody against the human protein microarray was actually not a false negative result. It was simply a misfit with the original protein on the microarray. So to summarize the study, what we did is we validated a mouse monoclonal antibody and a rapid polyclonal antibody against transglutaminase 2 and for this developed a three-step approach with protein array screening for antigen or cross-reactivity discovery, followed by multiplexed epitope mapping of the protein array hits for hit validation and epitope identification, and finally an epitope substitution scan to really narrow down the antibody response to individual conserved amino acid positions. Another part of the study was, or the goal of this study was to compare not only validation methods, but also the off-target binding of both antibodies. What we observed with the mouse monoclonal antibody was a clear target binding and few cross-reactions on the protein array, but absolutely no cross-reactions in the multiplexed epitope mapping on peptide microarray, and again a clear target specificity with an N-terminal epitope that was based on 7 highly conserved amino acid positions, what simply limits off-target binding. With the rabbit polyclonal antibody that was highly validated before by Western blot and immunohistochemistry, we observed a very strong cross-reactivity on the protein array and no target binding, what could later be explained by a misfit with the protein isoform on the chip. We observed a strong target binding as expected in the multiplexed epitope mapping, but also multiple weaker off-target effects. And taking the microarray platforms into account, the Human Proteome Microarray enabled definitely a broader screening. It gave a clear picture with a mouse monoclonal antibody, an apparent false negative result with a rapid polyclonal antibody, and a number of possibly false positive reactions that could hardly or only be limited to a limited extent be validated on the peptide microarray. The peptide microarray itself gave a clear picture with both antibodies with epitope resolution and in accordance with the expectations, but it does not enable such a broad screening as with the Human Proteome Microarray. And finally, the complementary epitope substitution scan enabled not only the identification of the epitope, but also the most detailed understanding of the antigen antibody epitope interaction based on each individual amino acid position, what also enables a cross-reactivity assessment of antibodies. So that's it from the study. And the data was presented in the ALPAF meeting a month ago. If you are interested to learn more about what peptide microarrays are also good for, we invite you to visit our website. There is an update sections with webinars and movies where we provided a 2-minute explainer video that shows a variety of peptide microarray applications for a number of different also serological approaches. And last but not least, if you'd like to get access to our products and services, we also invite you to visit our new online shop. So that's it from the data set. Now I'm open to your questions. Thanks for your patience.